Basic review of general microbiology. So prior to taking clinical microbiology, you've completed some type of microbiology course. And in that course, it went through the basic principles and procedures in microbiology. Now we're going to apply some of those basic principles that you've learned throughout this entire course, but it's really important that you remember those basic principles and have a great understanding before we move forward. So this is just going to be a brief overview of some of the main concepts that you should have taken from your previous microbiology course. So the first one, pretty simple. What is microbiology? Microbiology. So biology is the study of life. Micro is referring to microscopic organisms, also called microorganisms. It's studying life that needs a microscope in order to be able to view it. Now, although that's not going to hold true for all microorganisms, the majority of our microorganisms are going to require a microscope to be able to see them. Some microorganisms, like viruses, are acellular particles that are super tiny, so they're ultra microscopic. So they require specialized types of microscopes to be able to see them. Other organisms, like helminths or the worms, are not considered microorganisms, but since their eggs are, and some of their components are, we study them in the field of microbiology. But in general, microbiology, the study of really small living organisms. So there's prokaryotic microorganisms and eukaryotic microorganisms. In this class, we predominantly focus on the prokaryotic microorganisms, so bacteria. Once you take advanced microbiology, then we're going to look at more of the eukaryotic microorganisms, as well as acellular viruses and other related particles. But to start, we need to understand the difference between a prokaryote and a eukaryote. So a prokaryote, we can just call these bacteria for now. Very simple cell structure. There's no internal compartments or anything. It's kind of just one big open room in here. There's some genetic material that's folded up into a region of the cell called the nucleoid. And then there's some ribosomes and some simple structures. Prokaryote is talking about before the advancement of the nucleus or the development of the nucleus. So there is no nucleus. It's just a nucleoid-like region in the center of the cell that contains the genetic material. Eukaryotic organisms are going to have all of the common and familiar organelles that you would have learned about in an A&P class. So very similar to human cells, they're going to have the mitochondria, endoplasmic reticulum, Golgi apparatus. There's vesicles, vacuoles, a nucleus. In the center of the nucleus is the nucleolus, where ribosomes are assembled, and there's a lot of internal structures and details that provide specialized areas within this cell. So eukaryotic organisms have a nucleus, prokaryotes do not. And as I mentioned, this class is going to predominantly focus on those prokaryotic organisms. So when we talk about prokaryotes, or bacteria in this instance, prokaryotes also include archaea. But we're not going to talk about archaea in this class because archaea don't typically cause human disease. Now we may have some archaea in certain animal species, but they're not going to cause disease, so we don't really focus on them in a clinical microbiology course. Clinical micro, we're focused on the stuff that causes diseases, or the pathogens. So looking at bacteria, there's different cell morphologies that we can see with bacterial cells. A coccus is a sphere. A bacillus is a rod-shaped bacterium. Vibrio is a bent rod. Coccobacillus is kind of an elongated sphere or a very short rod. Spirillum is a spiral-shaped. And spirochete is a very long spiral. When you look at a sample under a microscope and you see bacterial cells, you should be able to identify what type of shape you're looking at. The next step in looking at cellular structure is looking at the difference in the cell wall. Now, there are a few species of bacteria that don't have a cell wall. They're specialized cell walls, like mycolic acid, and those types of specialized organisms we're going to talk about in advanced microbiology. In regular microbiology, in this clinical microbiology class that's going to focus predominantly on bacteria, we are going to focus on the gram-negative bacteria and the gram-positive bacteria. Now, gram-positive bacteria has a thick layer of peptidoglycan. Then they have a regular plasma membrane. And then the outside surface of that thick layer of peptidoglycan, you can find teichoic acid. This is just kind of a segment of the entire cell wall. So if this were to go around in a rod shape or in a spiral or a sphere shape, this is what the cell wall would be made out of. Gram-negative are different in that they have an inner membrane, an outer membrane, and on their outer membrane, it's part phospholipid and part lipopolysaccharide. And then they have a thin layer of peptidoglycan in the center. Now, this is important because we use a gram staining procedure to help give some preliminary information to a doctor about what's causing an infection in a person. So let's say someone comes in with, let's say, really bad neck ache, severe flu-like symptoms like a stiff neck, really sore muscles, and we suspect that it's meningitis. And we do a spinal tap or a lumbar tap, and we get some cerebral spinal fluid, and we send that cerebral spinal fluid down to the lab. Once you, the tech, receives that cerebral spinal fluid, the first thing you're going to do is put it on a centrifuge, and a centrifuge spins it around really, really fast in circles so that all of the hard, solid substances are going to go to the bottom of the tube while the liquid substances are going to stay at the top. Then you're going to get rid of the liquid and you're going to suck out some of that solid substance at the bottom to look for the presence of bacterial cells. Now, first we can just throw it on a slide and look, but it's better and gives more information if we actually go through the gram stain process. Now, we will be performing a gram stain multiple times in this class, but the basic principle is, first, you're going to fix the bacteria to a slide. And there's different ways of fixing a bacteria to the slide. You can either heat fix it, so getting it really hot and causing that bacteria to stick to the slide, or in the clinical setting, we more so do the methanol fixation. So you douse it with methanol. The methanol causes the bacteria to die and adhere to the cell or to the slide. Then the first step you're going to do is add crystal violet. Crystal violet is purple in color. So with both gram positives and gram negatives, the crystal violet is going to seep into the cell wall and it's going to form a complex with the peptidoglycan. So it's going to completely stain this and it's going to completely stain this layer of peptidoglycan. The next step would be adding the iodine. And iodine acts as a mordant, so it's going to allow that crystal violet, the primary stain crystal violet, to form a more tough complex and to bind a little bit more, a little better to that peptidoglycan. Then you're going to rinse with a decolorizer. The decolorizer can be acetone or 95% ethanol, and that decolorizer is going to wash away the purple. Now, when it washes it away, it's going to be able to wash away most of the outside purple on a gram positive, but because this layer of peptidoglycan is so thick, it will not be able to wash away all of it unless you over decolorize. If you sit there and you just dump more and more and more and more decolorizer on it, eventually all of the purple will go away. But in a normal gram stain procedure done correctly, only the top layer of that purple would go away. So this is still going to be purple after the decolorizer step. But if we look at the gram negatives, since this layer of peptidoglycan is so thin, when we do the decolorizer step, it is going to wash away all of the purple. So gram negatives, if done correctly, in the gram stain procedure, gram negatives should become clear after the decolorizer is added. Then the last step is the counter stain. And the counter stain is safranin. Safranin is like a red or pink color. So when we add the safranin, red or pink plus purple just makes it a prettier purple color. So these gram positives are going to stay purple. The gram negatives, since they're clear after the decolorizer step, they're now going to stain pink or purple. And then when you look at them under the microscope, you can differentiate the gram positives from the gram negatives based on what color they are. Again, gram positives are going to appear purple after the gram stain procedure, and gram negatives are going to appear pink or red after the gram stain procedure. So now that we have reviewed a little bit about bacterial cell structure, and you have an idea of what you're going to encounter in the clinical microbiology lab, now we're going to think a little bit more about what we're going to do with the bacteria that we do encounter. So, doctor or nurse suspects that there's an infection and swabs the patient for that infection and sends the swab down to the lab. So you, as a technologist or technician, you're going to evaluate that specimen and try to figure out what's causing the infection. And we do that by growing it. And we grow it so that we can identify it. And identifying it gives that information to the physician so they know how to treat it and how to get rid of it. So just a brief review of the 5 I's. So the 5 I's is how we grow bacteria: inoculation, incubation, isolation, inspection, and identification. Inoculation involves putting bacteria into media. So when we go to the next slide here, there's different types of media that we can use in order to inoculate the bacteria. So if we get a swab from a doctor, we're probably going to put it on an agar plate. So part of it's going to go on an agar plate. And that's so that we can grow a lot of what's in it. If we didn't put it in an agar plate, then we wouldn't be able to have enough bacteria to be able to make the diagnosis. So we have the agar plate, which is probably the most common media that you're going to use in the clinical setting. And then we also have broths, slants, and agar deeps. So agar deeps can be used to, if they're semi-solid, they can be used to determine motility or whether the bacteria can swim. And they can also be used for storing the bacteria. Slants can be used for storing the bacteria. Broths can be used for growing larger quantities of the bacteria that you've isolated. These are not very commonly used. A lot of times if we do get a swab from a doctor, we can mix it with some saline and then we can use that saline to dilute it across multiple plates, and then we'll streak it for isolation on those plates. The next one, incubation. Incubation is talking about holding the bacteria in particular environmental conditions to allow the bacteria to grow. So moving up to this one, we can control the oxygen in the atmosphere of the bacteria, and we can also control the temperature. And there's a lot of other things that we can control, but in the clinical setting, these are the 2 most important factors. So there's different organisms that have different requirements for oxygen. Not all organisms like oxygen, and a lot of our human pathogens are going to be facultative anaerobes. So if you go through this list, these should all sound familiar from your general microbiology course. I'm just going to provide brief definitions here so that you can review those definitions. Aerobic or an obligate aerobe requires oxygen. So people, like we are obligate aerobes. We require oxygen. If you cut off our oxygen supply, we can survive for like maybe a minute, but then we're going to have to take a breath of oxygen because our cells require oxygen in order to make energy. Bacteria, there's some bacteria out there that are also aerobic. They require oxygen. So when you incubate them, you put them in an incubator, you're going to put them in an ambient air incubator. Ambient air incubator means that it's the same type of air that would be out in our environment. Facultative anaerobe. Facultative anaerobe prefers oxygen but can live without it. So they can kind of do both. Aerotolerant anaerobe prefers to not have oxygen, but it can tolerate small amounts. So an aerotolerant anaerobe, those would prefer to be grown in an incubator that is supplemented with 5% carbon dioxide. That's going to be very important as we start to study our anaerobic organisms. In this class, when we start to incubate our Streptococcus strains, those are going to prefer to be in an environment without oxygen. Anaerobe, or an obligate anaerobe, requires an environment with no oxygen. It cannot tolerate any oxygen, and any oxygen will kill it. A microaerophile requires small amounts of oxygen for certain life processes. So we want to limit the amount of oxygen that these organisms are exposed to. Temperature requirements. There's some organisms that like to grow at room temp, like our molds are commonly going to be grown between like 25 and 30 degrees Celsius. Body temperature, the majority of our human pathogens are going to prefer to grow at body temperature. So that's between 35 to 37 degrees Celsius. Elevated temperature for isolation. So there's certain organisms that we can actually select for their growth by bumping up the temperature on the incubator. Like Campylobacter is one of them. If we were to grow a gram-negative organism and we grow it at 42 degrees Celsius and it's still alive, the likelihood of it being campy is higher. So we can kill off the other types of organisms just by cranking up the temperature on the incubator, and that will select for that species. Isolation. Isolation is talking about separating bacterial species from a mixed culture. So if we get that swab sent to us from a physician, and that swab came from a non-sterile body site, so in other words, like the swab was from a wound, and the wound was open to the outside of the body. The first thing we would do is inoculate it. We'd put it on some media. Then we'd incubate it. We'd let it grow for a while. Then we would want to try to isolate out the different species that are within that mixed culture. So going to isolation here, there's different ways that we can isolate. One of them is just by how we plate it. So we can plate using the streak plate method or the quadrant method, where we put the initial culture down on the tube, and then we're going to spread it from the first area to the second area, second to third, third to fourth, so that we decrease the concentration of bacteria that we're putting on the plate. Eventually, bacteria that land in the fourth quadrant will be in single cell form, so they will grow into individual masses of cells called colonies. We can also do quantitative isolation, so we plate a known quantity of the isolate that we have, and then we can streak it out and count how many colonies grow, and do a little bit of math and quantify how many bacterial cells were in that culture. Another method for isolation is selecting which media we want to use. So there's selective and differential media. Selective media selects for the growth of certain organisms while killing off other organisms. This plate, for example, is a biplate that we would commonly use for urine culture. On this side is just regular blood agar, and on this side is MacConkey agar. A lot of our pathogens that are going to cause UTIs, or urinary tract infections, that we would find in a urine culture are gram-negative. So if we give them media that selects for the growth of gram-negatives, we are selecting for the bad guys. We're selecting for the ones that we care about because we want to know which bad guy is causing the infection. Your normal flora, the normal bacteria that lives on your skin, has a lot of gram-positives in it. So if you get a lot of gram-positives in a urine culture, it doesn't necessarily mean that it's a pathogen. It could just be normal flora. So by having a plate that looks like this, a biplate, part of it will grow everything. The other part kills off the ones that are typically the good guys, and then we can see what pathogens this could be. Inspection. So inspection is looking at the characteristics of the bacteria to be able to tell differences and to be able to classify them into different groups. So going to inspection here, how do we tell bacteria apart? Well, we can use differential media to tell different species apart. So differential media will allow us to view bacteria that are different because they have different characteristics that show up on the plate. So if we have differential media like this one is blood agar, we can see that the blood agar differentiates bacteria from their ability to eat through the blood cells. So we have beta hemolytic, which can eat straight through the blood agar, alpha hemolytic, which can partially digest blood, leaving a green color or green halo around the growth. And then we have gamma hemolytic, which means that the bacteria do not have the ability to eat through blood cells. And we can see that on the plate. Like you can see here on the right-hand side, it has eaten straight through that agar plate. So this one would be a beta hemolytic organism. But other ways that we can inspect bacteria is by looking at what they can eat and what they can't eat. If we know that a known species of bacteria can eat these three substances, but it can't eat these three substances, and then we have an unknown bacteria that eats and can't eat the same thing as our known bacteria, we can start to make some correlations there and start to think about the identity of our microorganism or our unknown organism. So this is just a series of metabolic tests. I hope that the microbiology course you took previously, you performed some of these metabolic tests. We will be doing some of them when we get to looking at our enteric bacteria. But you can see like they'll change different colors depending on the different capabilities. So if they can eat a particular substance, they'll either produce an acid or they'll produce a base. And if they produce an acid or a base, that's a pH change. And there's pH indicators in these that can detect that pH change. The pH indicators will turn colors in the presence of an acid or a base, and that's how we can visibly see whether the bacteria can or cannot eat a particular substance. And then the last one, identification. So how do we identify which species is causing the infection? Well, we can correlate the gram stain with colony morphology and biochemical tests. So again, if we know that species A can eat these 10 things, and we know that our unknown, the one that we're trying to figure out, can also eat those 10 things, then we can say, okay, well, they eat the same stuff. We look at them on the plate. They look the same. They have the same colony morphology on the plate. And they also have the same gram stain result. So they're likely going to be the same organism, or at least very strongly related organisms. So we can do that way, and that's kind of the rudimentary way that we've been doing microbial identifications for many, many years. But now we have new technology, and that new technology allows us to use analyzers to be able to detect what species they are. So this is a picture of a Maldi, and this is in our actual microbiology lab. We are one of the only educational institutions in the United States that has a Maldi in their clinical lab. So pretty lucky to have this instrumentation. But this organism, or this instrumentation will take the organism and it will break it apart using a laser. And that laser will excite its ribosomal RNA, and the ribosomal proteins will be split into pieces and sucked up into a matrix, and they produce a characteristic kind of scatter plot and a characteristic read, so that we can accurately identify bacteria and even some yeasts based on the readout of this instrument. We can also identify using biochemical tests on different types of analyzers like antibiotic susceptibility analyzers, the AutoScan or MicroScan instrument. We also have the different types of analyzers that can read different color changes, like spectrophotometry can pick up on those, but the MALDI is a pretty accurate diagnostic tool. We can also go ahead and look at the genetic material. So we can do a lot of identification based on what genes are present and what fingerprint or genetic fingerprint is found in the organism that we're trying to identify. So that's how we would go through the stages of identification. It is our goal in the clinical lab to get whatever specimen the physician sent to us and figure out what's causing the problem. So it's kind of like going through these 5 eyes repeatedly to try to figure out what it is, and making decisions on what pieces of information are really important and crucial to get up to the doctors immediately versus what can wait until we get a full diagnosis. In addition to getting that diagnosis, we also want to be able to tell the doctor, how do we treat it? So how does all of this relate to clinical microbiology? Again, going through those 5 eyes, and then also looking at antibiotics. So hopefully in your previous microbiology class, you learned a little bit about antibiotics and how antibiotics have specific cellular targets that they go after, and how bacteria have developed the ability to elude antibiotics. So they have produced variations of the targets. They have produced chemicals that help pump the targets or the antibiotics out. They've produced different methods to be able to avoid getting killed or getting impacted by antibiotics, and that's called antibiotic resistance. So we need to be able to determine not only what's causing the problem, but how do we fix it? And we will be going through antibiotic susceptibility testing and a little bit more on that as we go through this course. But it's important that you review all of your general microbiology notes. A lot of the concepts I'm just going to assume that you know as we go through the course. I will do my best to do a little bit of review when there's something new, but a lot of this you should be proactive about your education in this course. And if there's something that you don't know, go look it up. Go back to your general microbiology notes, or go look on the internet or in the textbook. There's a free OpenStax textbook called Microbiology that's available for you to do some review and some reading. But all these concepts are going to continue to return. So we're going to focus on these and how they apply to the clinical setting throughout the course.